So let's talk a bit now about this process of transcription, right? Or essentially reading the DNA template and transcribing it into single-stranded mRNA. So there are three phases of transcription. I'm sure you've learned these in biology before. The first one is initiation, and then after initiation completes, we have elongation. That's where the majority of the nucleotides are synthesized. And then, as I gave an example above, at some point, the mRNA is going to dissociate from the DNA, and that entails termination of transcription. And that can be what's called extrinsic uh, or intrinsic. Sometimes the mRNA has enough information itself to actually dissociate from DNA. Other times it requires a factor called Rho. Um, so one big difference between DNA replication and transcription to RNA is that there is no primer required. In other words, we don't have to have a set of nucleotides that are laid down as this sort of initial nucleotides with a free 3' hydroxyl. In other words, you could say that RNA polymerase does RNA synthesis from scratch or de novo.